Excellent is your name, oh Lord. How excellent is your name, oh Lord. How excellent is your name. How excellent is your name. How excellent is your name, oh Lord. How excellent is your name. How excellent is your name. How excellent is your name, oh Lord. How excellent is your name, oh Lord. How excellent, Lord. How excellent is your name.
So hello members, we are back with our lessons which are online. Now today, as I already told you in our introduction, we are saying we are going to look at transmission of the impulse. This is under coordination in animals. So members, to understand properly coordination in animal, we have to know how the impulses are moving from one point to another. So members, to understand the proper transmission of impulse, as we have said, we have to understand the structure of the neuron and the nerve. When you look at a neuron is having features which are enabling to do the work. So it's having features like the dendrites. You'll see those branches as you can look at that other video. The dendrites are the ones which receive the signals, and they make a decision whether they can pass the signal if it is strong enough, and not to pass it if it is weaker, as you can see in that animation. Secondly, you will discover that the neuron is having what we call the axon. The axon is a simple fiber like a wire, which is conveying the signal or the impulse or the action potential from one part of the neuron to another. You can see that they are even what we call the terminal dendrites, which are supplying now the signal to the neighboring neurons. Now the cell body, you can also say cell body in that video, having the nucleus. You know properly nucleus is controlling the cell activities. Now when you bring many axon fibers together, now they will give you what we call a nerve. Nerves are many, they are propagating all over the body, and they are longer. You remember nerves, you can group them into the sensory nerves, the motor nerves, then what we are calling the relay nerves. So what I was saying that I think the structure of the neuron is clear now. Let us jump to what we call transmission of now the action potential or the impulse. To understand properly the transmission of the action potential or the impulse, we have already said that the dendrites are the ones which are going to receive the signal, and if the signal is strong enough, the dendrites will pass it via the axon to the neighboring neuron. Now transmission along with the neuron mainly depends on what we call the potassium ions and the sodium ions. Chloride ions are there, but we shall not put our focus on them. Now when you look at the distribution of potassium ions, as you are looking at that video, you will see that the sodium ions are represented in violet, and the potassium ions are being represented in blue. So after understanding the distribution of the ions, members, we are saying that one is going to take us to what we call the resting membrane potential. Remember, resting membrane potential is going to be a point where now a cell or the neuron has already achieved what we call the electrochemical equilibrium. Now achieving electrochemical equilibrium, mere hearing it may sound as if it is too complicated, but it's something simple across the membrane. Yet you try to follow up that video and then you'll discover what we are saying. Now to achieve electrochemical equilibrium, we are going to base on how many potassium ions are inside and how many sodium ions are outside. Then what is the magnitude of the positive charge outside and what is the magnitude of the positive charge inside, because of the potassium ions which are there and the negatives from the organic solids which are negatively charged. Now we are going to look at that video. We are going to discover that when the cell or the neuron is at rest, as already we have said it, that there are more potassium ions inside in addition to the negative ions. Then outside there are more sodium ions. This implies what? That we have more positives outside as well as the sodium ions outside. Then more negatives inside with fewer positives inside. Now the magnitude of the negative inside is going to become bigger. Then the magnitude of the positive outside is also going to become bigger. Now the difference in the concentration of the ions, be attentive here members, the difference in the concentration of the ions is what is going to be called the chemical gradient. Then the difference in the charges on the ions, this is what is going to become the electrical gradient or the electro gradient. Now when you combine the two concepts, the chemical gradient, then the electrical gradient, we are going to arrive to the concept, biological concept, which we are calling the electrochemical gradient. Now once you establish the electrochemical gradient, then we are going to say that the membrane is experiencing what we are calling the resting membrane potential. And now this one is measured in terms of potential difference. How are we arriving there? Look at the video. At the start we said more potassium ions inside in addition to the negative ions which are there from the organic salts and the chloride ions. Then outside more sodium ions. So you'll see in the video more purple outside, more blue inside. Follow up the outside side of the cell and inside. Then afterwards you are going to discover that the animation is showing you more positive charges outside. Then inside more negative charges. Then because of the more sodium ions outside and the fewer sodium ions inside, that one is going to take us to what we are calling the chemical gradient. Then more positives outside and fewer positives inside, but bigger negative inside, it's going to take us to what we call the electro gradient or the electrical gradient. And when you combine the two members, we are getting electrochemical gradient. The electrochemical gradient is taking us where? Is taking us to what we call the resting potential value of a cell. That is when the cell or the neuron is at rest. A neuron being at rest, don't forget that it doesn't pass any signal. It is natural. You can say dormant. No signal is passing through the fiber. That is the axon fiber. So at any time they can ask you to define the resting membrane potential. That you are going to talk about the potential difference obtained when the cell or the neuron has achieved what we are calling the electrochemical equilibrium. How do you achieve electrochemical equilibrium? That is when you have a bigger negative inside and a bigger positive outside. Who is bringing a bigger positive outside? The sodium ions. Who is bringing a bigger negative inside? The fewer potassium ions and the many negative ions from the organic salts. In addition, the fewer chloride ions which are being shown you in that video. So, when scientists tried to study the potential difference we are talking about of a neuron at rest, they discovered that it is negative 70 microvolts. So we are going to set on that value. On most of our graphs, you are going to see them in this video. They are setting a figure of negative 70 microvolts to depict the inner part of the neuron being more negative. And as we are going to reach a point of depolarization, we are going to see that that value will be lost, becoming more positive and positive. And as we shall talk about repolarization ahead there, the value you will see that is becoming more negative, back to the natural value. And many other words will come on: hyperpolarization, overshoot, refractory period, absolute, and so on. So, guys, you have already seen that the natural PD of the neuron is negative 70. That is when, remember, we have a bigger negative inside and a bigger positive outside. The bigger positive is as a result of the many sodium ions outside, don't forget that. But again, the bigger negative is as a result of the organic salts and some chloride ions with a few potassium ions inside the membrane. However, the PD doesn't remain negative 70 forever. Remember, the neuron its work is to allow the impulse or the signal to pass. So as the signal is going to pass, we are going to see that the membrane of the neuron is going to allow some ions which were outside to move inside, and those ones which are inside to move outside. Now, the ions are going to pass via what? The ions will pass via proteins which got embedded in the cell membrane. Remember, when you go back to the fluid mosaic model, we talked about the intrinsic and extrinsic proteins. Now, those proteins, some of them are channel proteins that can allow materials to enter and to come out. Now, we are going to look at the channels. You will discover that in that video you have different channels being shown to you. For example, the potassium channels, the sodium channels, they are all among the channels being illustrated in the video. Now, those channels, some of them open as a result of high voltage due to the stimulation of the membrane, like the potassium and the sodium channels. But others can only open when there is a molecule attaching on them. The videos showing them as a ligand conjugated or the ligand-gated channels. Then when you look at others, they only open when the membrane stretches. The purple beads moving outside and then the two blue beads moving inside in the video. This implies that there is another mechanism which can enable these ions to move naturally inside and outside. And this one is not another idea or another mechanism apart from what we call the sodium-potassium pump. Now, to regain the natural electrical neutrality or the potential difference of the membrane, we are seeing that here the potassium-sodium pump is going to act. This one is using ATP. The ATP or the energy stored in as ATP is going to allow 3 particles or ions of sodium to move out of the membrane as 2 potassium ions are moving inside. So when you follow up our video there, you will see that now the 3 purple ones are moving outside and the 2 blue ones are moving inside. So that is what we are calling a sodium-potassium pump. What is the role of a sodium-potassium pump? That one is now to make a cell to regain its PD of negative 70. Now, what is important to note, how the length increases or the pressure changes. So this one is as a result of the physical or mechanical factors that can cause them to do what? To open. So we have the channels and they open different. Now, our area of concern is that depolarization. That depolarization will take us to what we call action potential. That is now allowing the impulse or the signal to pass. Now, when the membrane becomes stimulated, remember, as what we see, the stimulation, if it reaches the threshold value, the sodium channels are going to open. When the sodium channels open, as I've already seen, we are going to see that more sodium ions are going to influx in. The video is showing properly, violet or purple, moving inside via those burger-shaped structures, which we are calling the protein channels now. Then as they enter inside, we are going to see that the inside of the membrane becomes more positive. Remember, we said naturally it must be negative. Now it is becoming more positive as a result of the influx of the more sodium ions inside. Then at the same time, we are seeing that the potassium channels for them remain what? Closed. Now, because of the entrance of the many positive ions inside, we are seeing that the potential difference of the membrane changes. It becomes more positive. It raises upwards from negatives to positives. Now, it will raise up to around 0. As the graph is showing you in the video, it is raising up to around what? 0. Now, when it shoots above 0, that one in biology always call it an overshoot. At that time now, the channels of sodium, which were allowing sodium to enter, again close. because they are going to become inactivated because of the overshoot. Now, because of the channels becoming inactivated, we are seeing that no more sodium is entering. And now we are seeing that the process of depolarization has been achieved. Now, if the depolarization is big enough to reach threshold value, we see that a generator potential is generated, then action potential, which we are calling an impulse. After firing, the video is showing clearly there. After firing an impulse, we are seeing that the cell must regain, or the neuron must regain its natural neutrality, which is natural PD. How is it going to regain it? We are going now to come to a second process, which we are calling repolarization. Now, as the sodium channels are inactivated and others closed, we see at the same time the potassium channels or the potassium gates are going to open. When they open, the potassium ions which are inside the neuron are going to move outside. As they are moving outside, they are reducing the positive which was being created by the entrance of the sodium. And then now we are seeing that the positive is lowering to become now a more negative. As more potassium moves out, the membrane continues to become negative. However, we see that there is always a delay in the closure of the potassium channels. Because of the delay in the closure of the potassium channels, we see that the negative again become a bigger negative than the negative 70 which we expect. It moves to negative 60 downwards. Now this one implies that the inside part of the neuron is becoming more and more negative, and that condition where it becomes more and more negative, that is less than negative 70, that one is going to be known as the hyperpolarization. Now, from there, we are seeing that during the process of repolarization, the neuron is experiencing what we call the absolute refractory period. That is when now the neuron cannot fire another impulse or another action potential. Why are we seeing that? We are seeing that the sodium gates now have closed because of the overshoot we talked about. And we are seeing even others are being converted. The inactivated ones are being activated now. Now we are seeing that even though the sodium channels would open, there is no chance of the sodium ions again influxing in. That one was not a mistake. It was a good design now to cause now a delay in the firing of the second impulse after firing the first impulse. This one is important because now it causes a delay in the firing of another impulse. It prevents firing of an impulse immediately after the other one has been fired. Then secondly, it prevents the backward flow of the action potential or the impulse along the neuron. We want to go in the forward direction. We call it propagation. It must propagate, not again propagating backwards. So take a note of that, why there is now the instance of the absolute refractory period. That we are saying that is a period when there is no firing over an immediate action potential after the neuron has already fired or invoked a signal. Then we have what called the relative refractory period. That one is going to be now experienced under what we call the hyperpolarization. So after absolute, then we are seeing now refractive. Refractive, remember, with the other one which is absolute, you will find that the challenge is that it is very hard to generate a stimulation which is unusual to make now the gates to open again, the gates for sodium to enter, as more sodium ions are still inside. Now, during the process of hyperpolarization, we are seeing that more potassium ions have moved out now. The bigger negative, the gates are still open. Now, you need to first now prevent the potassium ions from going outside, because now the cell must regain back its natural PD to begin to start up a new phase of firing an impulse. But now we are seeing here it is already having a very big negative, which very big negative must first be leveled now back to the natural point before you fire second you fire the second action potential. So now we are seeing that period when now the cell is regaining its natural PD. That is when now we are going to see refractory period. what we are calling the relatively failure period being what? Being experienced. Now, after firing an impulse, remember, the firing of the impulse we have talked about, we have based mainly on the depolarization. When the more potassium ion, the more sodium ions have entered, the membrane is going to become depolarized. Then depolarization, the membrane is regaining its natural potential difference, or the electrochemical gradient. Then hyperpolarization, we are saying the potassium ions have moved more outside because of the closure of the potassium channels. So you have to understand the polarization, and hyperpolarization. Depolarization, more sodium entering. Then the polarization, we are seeing that more potassium ions are moving out. Sodium ions are entering because of the opening of the sodium gate. Potassium ions are moving out because of the opening of the potassium what? Gate. Now, hyperpolarization is as a result of the delay in the closure of the potassium. You are going to look at that video, you will see as the potassium ions are moving out, and when does the sodium ions move what? Move out. Now, from that point, after firing the impulse, we are seeing that the speed of the impulse propagating will depend on what you call the positive feedback mechanism. The different segments will become depolarized for the impulse to propagate from the left-hand side, the right-hand side of the axon fiber. Now, from that point, we can also talk about the speed at which the action potential is moving along the axon. That one is mainly controlled by different factors, but the major factor is going to be now the presence of the Merone's teeth. When you look at that video, you will see the swellings along the axon, covering the axon. Now, the more the covering, which you are calling Merone's teeth, the faster the conduction of the impulse. So you will talk about, when you look at that video, you talk about what we call salutary conduction. We are seeing that the action potential is having the ability of jumping from one node of labia to another node of labia. The video is clearly showing that light from one joint to another what? Joint. Each swelling is having a gap. Between the swelling and the swelling, there is a gap which are calling node of labia. Now the action potential, the impulse, or the signal, is being shown that video jumping from one node of labia to another node of labia. That is how now the speed is going to increase. In others which are lacking the Merone's teeth, that kind of jumping or conduction doesn't occur. But because here we have the teeth from the Schwann cells, we are seeing that now the flow of the action potential or the impulse is very, very what? Rapid. So therefore, if they ask you those ones, the neurons which are myelinated and those ones which are non-myelinated, you'll discover that the myelinated ones, they have a faster conduction. Then the non-myelinated one, they have a slow conduction.